Hi, everyone.
I'm Lisa.
And I'm Steph.
And together we are
PD Connect. Thanks for joining us today. We are PD Connect. You can check us out at pd-connect.org. We teach exercise and education for people with Parkinson's. You're at our speaker series, and we've got a really great speaker here today with us.
Yeah, we are a small nonprofit in the San Francisco Bay Area, but as we like to say, virtually
wherever you are.
Well, anyway, so today, every first Monday of the month, we have a guest speaker. And today we are very honored to have Dr. Karen Raphael. And she's going to talk about hope or hype, how to look beyond the headlines reporting PD breakthroughs.
Oof,
so let me tell you a little bit about Karen. So Karen Raphael is a Ph.D. psychologist and chronic disease epidemiologist. She currently holds the title of Professor Emerita at New York University, NYU. During her 40-year academic and scientific career, Karen was principal investigator on multiple NIH and foundation grants designed to advance understanding of medically unexplained pain. Well, Karen was a care partner for her father when he developed Parkinson's disease in 1996. She received her own Parkinson's diagnosis in 2010, months after acute symptoms appeared during her failed trek to Everest Base Camp in the Himalayan mountains. Yikes, just to think about going to Everest is already like, whoa.
That's incredible.
In the 15 years since her diagnosis, she increased her already rigorous personal exercise regimen by following the research evidence and increasing her workouts to 3 to 4 hours daily.
Wow.
Yes, you heard me correctly. 3 to 4 hours daily. I love it.
Give her a high five.
Me too. I just want to like be one of those little, you know, the little ducks that follow in a, never mind. Especially since her retirement in 2021, Karen has used her advanced knowledge of epidemiological and clinical research methods to help people living with Parkinson's disease to better understand relevant clinical research literature through delivering lectures and webinars like this, and by serving as administrator for 2 research-oriented Facebook PD groups. Karen currently reviews grants for several PD-related organizations and provides short-term gratis advisement and counseling for people recently diagnosed with PD. She also serves as a patient research advocate for the Parkinson's Foundation and as a global ambassador for PMD Alliance. Karen lives with gratitude every day. And we are very grateful to have and to host Karen. So thank you so much for being here.
First of all, thank you so much for the invitation. I really enjoy speaking to my peers, and you are my peers. I don't know how many of you have lived with Parkinson's as long as I have, but it's been a long time, and things have changed. So if I, in the course of this presentation, if I have trouble finding a word, if I stumble, if I seem disorganized, I blame it on the Parkinson's. If I say something brilliant and inspirational, it's all mine. I'll own it. So, you know, take it whatever way you want. I will do the best I can. And I've actually responded to, Lisa had sort of said, well, topics come up sometimes in discussions, and then people ask about certain things, and you've discussed certain things in the past. And I can give you my own take on those kinds of issues. I'm happy to do so. And it's one woman's opinion, but it is one woman who has a fair amount of research experience. But again, I'm one person. So take that as you like it. Okay. Okay. So the other thing is, although I should be doing my loud therapy more often and practicing more often, sometimes it's hard with everything else that's going on. Sometimes exercise doesn't include my vocal cords maybe as much as it should. Now, all right. So anyway, so you know, I've been around a while, and really want to talk about the kinds of things that we might hear about and claims that people are making, and what's the nature of the evidence, what's the strength of the evidence. And I will try to speak louder but if it is too soft for you, somebody just, give me a little punch and I'll try to make it louder. Okay. So where are we going to see or hear about these kinds of claims we're making? Hold on a second. Certainly, we hear word of mouth claims, all right? I mean, we may have our own experience, our own personal experience, which informs our beliefs about what may be really helpful for living with Parkinson's, which may help our symptoms or may even be disease modifying. And we'll talk about that difference in a minute. There's also what I'd like to think of very loosely as word of mouth, all right? That is people we know tell you, oh, I met somebody who had this experience. Do you know anything about it? And how do you take that? One of the big sources of information, right or wrong, is social media. Okay? And, when you start seeing things that say things like a treatment is groundbreaking, guaranteed, revolutionary, the secrets your doctor won't tell you about, all that kind of stuff. Those are buzzwords I tell you, this is probably, well, I guess I can say BS. All right. That's hopefully that won't get beeped out or something. The other thing that people tend to think is more useful, but may or may not be, are headlines and articles that are titled or written by science journalists. Any of you who are familiar with like Parkinson's News Today or some of these kinds of news services about things that are happening in Parkinson's research, not just that, Parkinson News Today does other things besides that. Some of them are good, some of them don't necessarily interpret science correctly because it's not that easy if you don't have that specific science background. Some of you have particular bloggers or vloggers that you're used to looking at and you really trust them and you understand what they're saying and how they're saying it. That's another source of information. I would say whatever you think about some of the PD charitable organizations in terms of what their priorities are or whatever, they tend to be better than the or what do I say, the bloggers or even the social media posts in terms of giving you some general background information about something you've heard about. It may not necessarily include the newest information out there, but it is a place where the information that's there is generally reliable. And the other place that I can go to that you may not have such easy access to are peer-reviewed journals. This is where you see the original scientific articles published. These are peer-reviewed, and people will tell you that some of that stuff is incorrect. Things get withdrawn all the time. So even what is supposed to be the highest standard of evidence, there are errors that occur. And that's why science requires replication. So when one study comes out, it then gets translated through all these upper methods, through, from peer-reviewed journals all the way back to word of mouth. Some of that's fine, okay, but sometimes those peer-reviewed journals are erroneous, okay, the conclusions they reach, even with peer review. And peer reviewers are typically not paid, all right? So we do this, or I did it when I still get asked as an Emerita faculty member to review for lots of journals. I can't accept all the requests I get because it's done for free, right? So, and some reviewers don't do a careful job. So sometimes mistakes happen and sometimes people fake data, right? That's the sad reality of the world we live in, okay? So when I say people making claims, these breakthroughs or whatever, what kind of breakthrough claims? About what? First, we're talking about treatments, certainly. Treatments that reduce PD symptoms. They could be motor or non-motor, often cognitive. And it could be about medication side effects. We're talking about, let's say, dyskinesias. Are you familiar with those kinds of terms? Are most of the folks in your group familiar with dyskinesias, would you say? Okay, great. Okay. And I separate from that disease-modifying treatments, that slow progression. And somebody once actually in that Parkinson's research interest group asked a question very recently that said, well, what's the real difference between things that reduce symptoms and things that are disease modifying, why do we need to distinguish the two? We do need to distinguish the two because Parkinson's is a progressive condition. So we're interested in not just things that will deal with our immediate symptoms, but things that can maybe help slow the progression of symptoms. That is a much more powerful type of intervention. And we're certainly interested, I would hope, in looking at people who don't yet have PD, but might be at risk of developing it. And we're talking there about what, as a public health person, I would call primary prevention, that stop people from getting Parkinson's, because you might have heard about the projection of a Parkinson's pandemic, that the volume of people, the number of people, is not just going to stay flat. We're going to exceed the number of people with Alzheimer's. Okay. So it is, and even more recent than some earlier articles came out declaring this Parkinson's pandemic, are things that are now newer projections that are coming out saying it is even more severe than we originally anticipated. And it's not just due to the aging of the population, it is due to more exposures to what I'll call risk factors, or things out there that increase risk of developing Parkinson's. And again, there are claims about, this is something Lisa asked me to speak about specifically, these new diagnostic methods to identify those who are most likely to either develop Parkinson's, or I would say to confirm a current diagnosis beyond the clinician's best guess. So let's start with what I think is the most basic kind of information that we are likely to get, and we'll sort of move up the chain, all right? So the challenge of interpreting personal testimonials about how well a treatment helps PD symptoms, or even interpreting your own personal experience. Now, let me just back up for 1 second. So I have exercised like the devil for 15 years with Parkinson's, and very hard before that for all my adult life. I'm not an athlete, but I know that healthy aging requires being active. And if I don't have a chance to say it later on, I think many of the lifestyle factors that are associated with healthy aging are also factors that are associated with lesser progression or more gentle progression of Parkinson's. That is not at an individual level, but at a group level. So I can't tell you exactly what your course is going to be if you exercise 3 to 4 hours a day. I can't tell you that how well I am doing is directly a function of my exercise regimen, possibly based upon the research, but I can't be sure. Oops, sorry. And where did these challenges come about, and why can your personal experience be misleading? First of all, many of you have heard of the placebo effect, correct? You've heard of it. Now let's talk about what it really is, right? It's not something that's fake, right? It's not just belief, but it is a perceived or actual health benefit that's caused by the expectation of benefit. And it's really associated very often with learning, right? So that, for instance, if we have learned that, I don't know what color your carbidopa levodopa, assuming you're taking it, is, but if you get another pill that is the same color as the one that you're used to taking, which you know increases your available dopamine in your brain by converting levodopa to carbidopa in the brain, and then somebody gives you another pill the same color and says, this is going to improve your dopamine levels. You take that. What do you think is going to happen? Your dopamine levels will actually increase. That's not fake, okay? That's not just expectation, but it's based on your prior experience, okay? And I said that the placebo can cause actual changes in dopamine that are comparable to therapeutic doses of levodopa. And we can find this out and verify this by using various kinds of functional neuroimaging. You might be familiar with some of these. like PET scans, functional MRI. These are things that don't just look at the structure, but really look at the function of the brain. And the one thing that usually is used to differentiate placebo effects from what is called real or direct effects or specific effects of an intervention is that placebo effects don't tend to last very long. At most, at very most, maybe 6 months. But they might reduce symptoms, but they're not going to affect disease progression. That's a general consensus in the field. And here, this is something that was written by someone who I think of as the father of the biology of the placebo response. And as a pain researcher, I certainly did a lot of studying about placebo responses. And Benedetti, who is an Italian guy, wrote quite a long time ago that hard science tells us placebos can reduce symptoms like pain and muscle rigidity in PD, but the progression of the disease is not affected. For example, in Parkinson's disease, the neurons keep degenerating even though some symptoms can be reduced for a short time. So Benedetti used Parkinson's as his model to study the placebo effect, which I think is kind of cool. Okay, another reason why personal experiences can be misleading. I'll start off by just calling it variability in course of PD over time. And I'm going to borrow something that Lori Mishley, who some of you are familiar with, I know that Lisa is, and she is a naturopathic physician outside of Seattle or in Seattle, who combines her practice is specifically for people with Parkinson's. She actually consults all over the country with people who can't reach her in Seattle. She also has some research training. She has, I believe a master's of public health and epidemiology. So our backgrounds overlap there. And she did some very interesting research. One of the things, this is something that I borrowed from one of her earlier publications. This is using a scale that she created called the Pro-PD, which is really 33 different symptoms. In this scale, the higher the score, the worse you're doing. So if you have a low score, you're in the excellent range. If you have a high score, you're doing poorly, okay? So as we move up, we're doing worse. But if you look at time since diagnosis, in general, as shown by this line right here, over time, people do worse. But what's really important to notice is the variability. So let's look at 15 years where I am now. Well, I don't know if I'm doing excellent, but I'm probably somewhere between excellent and good. I don't know. Depends upon the day, depends upon how my meds are working, okay? But there are plenty of other people at 15 years out who would be doing and would based upon their scores on the Pro-PD, they would be considered to be doing poorly. So when I look at my experience and say, well, I'm doing pretty well for 15 years, I don't really know how I would be doing without the exercise. Would I still have been over here? Would I have been over here? I don't really know. So it's very difficult to say, I'm doing great. It's due to X, Y, or Z that I've been using or taking, or this nutraceutical I've been using, or this new kind of exercise I've been doing. It's very hard to infer what causes our current symptoms, okay, or improvement, or increase in symptoms, on the other hand. Okay. So we really can't know what our symptom changes would be like if we weren't using the new treatment. Okay, another reason why personal experiences can be very misleading are the problems of diagnosis. Now, in particular, and I'll get back to this later on, when somebody tells you that they were cured of Parkinson's by some particular treatment, and you see this particularly on Facebook and some of the other social media sites, people say, I am cured. I took this particular supplement or whatever, a group of supplements, was treated by this particular practitioner, and I don't have any symptoms anymore. I would question that, and I'd have to ask, really correctly diagnosed with a progressive neurodegenerative condition of Parkinson's disease. And I think that's a question that is a legitimate one, because we know that misdiagnosis is not uncommon. It is common. And here's just two studies, but particularly with the outcome being, or the gold standard being, a brain biopsy after death, where you can really see what kind of neurons are missing in the brain, where you can look for certain abnormalities, certain clusters of things that we'll talk about later. Somewhere between 10 and 20% of people who are clinically diagnosed, even by a movement disorder specialist with Parkinson's disease, using a standardized algorithm, may not really have Parkinson's disease. They may have a Parkinson's lookalike. They may have one of the Parkinson's plus conditions that can even be more severe, like progressive supranuclear palsy or multiple systems atrophy. And these are kinds of things that over the course of time, those progress more quickly. And Lewy body dementia in the first year, basically, if you develop increasing dementia in the first year after developing motor symptoms, they say you have Lewy body dementia. If you don't, and it happens later, they say you have Parkinson's disease with dementia, right? It's kind of arbitrary, but that's the way they differentiate the two. But the progression and how long you can live independently really varies based upon what diagnosis you have. And if you have, idiopathic Parkinson's disease, let's call it for now, you truly have Parkinson's disease that's sort of like most other people who have been correctly diagnosed, you probably won't have any significant shortening of your lifespan. But with some of these Parkinson plus conditions, you do have much quicker progression and shorter lifespan. So when somebody says they were cured, you say, well, did they really get the right diagnosis? Are you hearing me okay these days, right now? Yeah, it's okay. Yay. Okay, thank you. Okay, I'm going to give you take a couple of, I'm calling them case studies, but they're things that I've seen in the literature to sort of bring the point back home. Anybody ever hear of Manitol? Or you're not old enough to, okay, I see Lisa shaking her head anyway. All right, so Manitol, I think maybe it was about 10 years ago that claims started to come out about this thing called Manitol, which looks like this. It's really a sugar alcohol. Okay. First, there is a personal testimonial, which is actually published in a journal, not a high-ranking journal, but it was still published by this man, Don McCammon, who also used to sell, or maybe even still sells, a variation of mannitol that I believe he's patented. And it's easier to digest. A lot of sugar alcohols upset your stomach, especially in large quantities. So he had his own version, which he said was easy to digest. And he also made the claim in this journal that I'm 66 years old, had all the symptoms of Parkinson's. I started taking mannitol orally, and after 30 days, I could stand and walk regularly. That's an impressive claim. Well, I looked at this write-up, and I wrote him. And I said, Don, can I ask you how you were diagnosed? And he said, well, I really never got a diagnosis. I had a brother who had Parkinson's, and I saw that I had some symptoms similar to him, that I was, tripping and having some trouble with walking. So he self-diagnosed Parkinson's. So maybe after 30 days, he could stand and walk regularly, but he never really had the diagnosis. You've got to watch that. And I said that mannitol is a low-calorie sweetener, and it actually has a medicinal use. It can actually reduce pressure around the brain, right? And grass is generally recognized as safe, right? So people don't think they're tremendous, or scientists or the FDA doesn't think there are major problems with it other than in large quantities upsetting your stomach. But you see McCammon's claim, and then there started to be some science around this stuff. In vitro studies, those are test tube type studies, okay, petri dishes, as well as lab studies in fruit flies and in mice. Okay? And those studies are particularly interesting because they found that fruit flies who were, I think they were genetically modified so that they had trouble flying, and had trouble even climbing the sides of a jar, a glass jar. When they fed the Manitol, they could climb and fly better. And mice who were had induced Parkinson's disease by giving them a neurotoxin that destroyed the part of the brain where the dopaminergic neurons are. That's the substantia nigra. So they basically had a one-shot kind of poison or toxin administered to them, and very quickly that part of their brain was destroyed. So it's not like humans, but they found that when they fed these mice or these fruit flies Manitol, their symptoms reduced and they started to appear more like fruit flies or mice that hadn't had these disabilities inflicted on them. So that seemed really promising. But now Manitol is not the kind of thing that big pharma is going to be interested in, right? Because they're going to be interested in something that you can patent. So an Israeli entrepreneur started to get really frustrated about this, that there are treatments that might be efficacious, at least look, based upon the basic science stuff that was out there and the animal studies that were out there that looked like they might be efficacious. So he created a platform called ClinicRound. Anybody hear about this platform? It was a few years ago. All right. Anyway, it basically was a sort of it was a web-based survey instrument in which if you were willing to try Manitol, which you could probably, I assume you can get in, you can either get it on Amazon from Don McCammon, or you can probably get it in a lot of pharmacies or whatever, as it's, you know, or even supermarkets for that matter. So he created this survey instrument in which people who were willing to try Manitol reported that they were starting it, and then they continued over time to report their symptoms. So that was the model for ClinicRound. The idea was this is not going to be equivalent to a clinical trial, but it might show what the interest of patients was out there, and it might provoke enough interest by science so that eventually a clinical trial could be run. And yeah, he called it citizen science or undone science. The idea is that it's going to look, it was started to look at Manitol and Parkinson's, but could be expanded to other conditions and other kinds of treatments. So they eventually, at some point, I think they reported that 1,500 people reported their experience with Manitol, at least their initial experience. But when I looked for people who had used Manitol for 6 months or more, there were only 78 people. Okay, of those 78 people, 56% said they improved, 7% said no change, 37% said they got worse. And I was like, I don't even know how to interpret that if only 78 out of 1,500 people used it for that long. Nevertheless, there are 1,500 people that started out being interested in Manitol. So that did lead to Hadassah Hospital in Israel again to fund some investigators to do a small double-blind randomized clinical trial, and we'll go through exactly what that means again in a little bit. And they found that the efficacy was no greater than placebo. So we go from these personal testimonials to some preclinical research to a double-blind randomized controlled trial. We reach very different conclusions about the efficacy of Manitol. Okay. Now I'm going to give you one more sort of personal experience shot. And I found this interesting because it's been there on YouTube for a long time. And this gentleman is a British guy named Ian Friselle, and he says that this video, well, this is a short portion of the video clip. Oh, I've got to get that taken off in order for this to work. Sorry. Let's see if I can not talk about. And I should say that he's a person that's had deep brain stimulation, so he doesn't currently have symptoms. But if you've had deep brain stimulation, you can turn off the stimulation and you'll see what you look like or what your symptoms are like without deep brain stimulation. So he did a video of himself with, first you can see what his tremors were like, quite intense, okay, when didn't have the stimulation from deep brain stimulation on. And he was particularly interested in marijuana. And he then, okay, here are his lovely tremors, not so lovely. And here he is, dramatically changed after he medicated with, he says, 30 milligrams of marijuana. And it was, I've had people share this with me and say, isn't this impressive? Have you tried cannabis? I mean, I have, but mostly for sleep, but I have a few things to say about it. But basically, it is one man's experience. And, you know, at first I looked at it and I said, well, I don't even know that he smoked marijuana in between when he had the tremors and when he didn't. So even though some people might think it's a dirty word, I am a scientific skeptic. All right. And that makes me say, what could be wrong with this? Isn't necessarily wrong, but makes me wonder whether there might be better evidence. And because marijuana has been decriminalized or made legal fairly recently in most arenas, and certainly in the US, where a lot of research of all kinds takes place, it's been very, very recent and still difficult to do studies on marijuana or marijuana products or and what their clinical utility might be. And this is one study that I saw, which is a randomized, double-blind, parallel group, placebo-controlled, single-site study that was published very recently. And they looked at a combination of CBD, which is a very common, I think they call it a terpene, and THC, which is the psychoactive component that makes you stoned. So a combination of that in, I believe, some kind of oil. And then a placebo that was also an oil, but didn't have any CBD or THC. And they found that it did reduce the resting tremor more than the placebo, the active CBD THC. But there's a problem. Sleep, cognition, and activities of daily living, I say, showed a treatment effect favoring placebo, or another way to say that is people were doing worse if they received active CBD THC. So we've got a problem that it may be helpful for your tremor, but it certainly has side effects. So it's a real mixed bag. There certainly needs to be a lot more research done on this. And I think there's, you know, a call for a lot more research to be done on cannabis. Now let's move to something that Lisa asked me to talk about. And I'll try to frame this within how we're talking about, we're not talking about personal experience here. Nobody's taking well, people are personally taking, you know, undergoing stem cell treatment. But let's talk about what we know about stem cell treatments and move from preclinical research to potential FDA approval and where the promise might be, where the hype is, and where the hope is. Okay, so I'm not going to get really specific about the different kinds of stem cells here because I think it's way too much detail for us to all remember, including myself, right? But basically, these stem cells are implanted into the brain to take place of cells that have been lost. And you see headlines like this: Parkinson's patient moves freely again after world's first implant of lab-grown cells in his brain. Dramatically positive. Okay, makes you think, oh, I want stem cells. And that's probably why you see all these commercial offerings in the US and all over the world. Okay, and they're expensive. If you have to pay for it, it's not research, because they wouldn't charge you if it was research. Sometimes they'll tell you it's research. And also, if you see that they're offering treatment for multiple conditions like Alzheimer's, MS, you name it, migraine, as well as Parkinson's, be highly suspicious. The reality check And there is currently no FDA-approved stem cell therapies that can stop or reverse Parkinson's. And I'm going to travel, I don't know how else to transition to this, but I want to give you just an overview of when we're talking about preclinical and clinical research, where this stuff all fits in, and then we'll return after that to talking a little bit more directly about what's going on with stem cells. Talk about clinical trials and clinical research. Yeah, at the beginning, before we get, this is really not clinical trials. First, you usually start off with animal laboratory studies. Then you move into these various kinds of clinical trials. We start, oops, sorry about that. Nope, sorry, okay. Okay, so let's start off with the preclinical and look at the lab studies for stem cells. And here was one quote that I saw out there that I started reading. Honestly, in my wildest dreams, I couldn't imagine a single therapeutic having this much benefit, says whoever this person is. I really think this will be a game changer for the clinical management of Parkinson's disease. Are you folks hearing me okay right now? Yes? Okay. Thank you. Okay. And this Dr. Brigitte Schul, who's director of gene discovery and stem cell modeling in another country, developed a model that turns skin cells into stem cells and coaxes them to become dopaminergic neurons. That's basically what happens when you're doing this. It turns out that these were created by Parkinson's disease in a culture dish. Right? And that's what led to these wildly speculative claims about this being a game changer. Okay, useful perhaps to explore the underlying cause of Parkinson's disease. Let me get this thing back on. The underlying cause of Parkinson's disease, but really too soon based upon the Petri dish study to think that this is going to be a game changer. Okay. Again, preclinical research, not with humans. Now, probably what there's a lot more out there that you see and hear about are the animal studies. The first thing to remember when we see animal studies is that animals other than humans don't get Parkinson's. They get it in the lab artificially, like exposing them to neurotoxins or genetically breeding them in a certain way. And these are usually very different than Parkinson's disease in humans. So we, and to a lesser extent, our closest primate relatives, have dopaminergic neurons. These are the ones that we lose, these nerve cells. They're coated with this black substance called neuromelanin. You don't see that happening in anybody else, any other animals except our closest primate relatives. So if you start looking at mice or fruit flies, their dopaminergic neurons are very, very different from ours. And these we have certain dopaminergic neurons that don't have neuromelanin on them. But the ones that have neuromelanin, this black substance, they're very susceptible to any kind of abnormal stress in the brain, and they'll die very quickly if there's any kind of unusual, what they call oxidative stress. And I would say, unfortunately, that animal studies are our largest source of false hopes and false headlines. We see an animal study that looks really promising. We get all excited. The translatability of animals studies to humans, it's really not good. It is basically batting zero. If animal studies translated to humans, we would have cured PD dozens of times. So human brains are unique. I think there was perhaps a report of one single individual, a marmoset, I believe, that looked like they might have Parkinson's, but we're not even really sure. So again, I wanted to talk about the different stages of within clinical trials. First, there are phase 1 studies. people sometimes don't even think of them as clinical trials. These are basically safety studies, dose ranging studies to figure out what might be a toxic dose to humans. And they're done not necessarily even in patients, but maybe given to healthy volunteers sometimes. Sometimes they're given to patients. But it's really to set a very low bar of just showing that this is safe to study further. And it's only later, after things have passed phase 1, that we move to phase 2 and phase 3 studies. Typically, we start with a small phase 2 study, and then assuming things look okay there, we then move to what's called a phase 3 study. Now, if this is an intervention that is seeking FDA approval, whether it's for a drug or a new device, you do the phase 3 study or studies, then you go for FDA approval. And then sometimes after that, when they're looking for long-term adverse events, there might be a phase 4 study. We're not going to really talk about phase 4, but I'm particularly interested in us focusing on the difference between phase 1, 2, and 3 here. And these are all considered clinical trials. So what's special about randomized controlled trials? Now, if they're well done, a randomized controlled trial allows for the strongest evidence about whether a treatment is likely to improve symptoms or quality of life for people with Parkinson's. PWP, sorry, that's my common abbreviation for people with Parkinson's, okay? And people with Parkinson's are randomly assigned, kind of like a flip of a coin, to 2 or more potentially helpful treatments or a comparator treatment, which might be standard treatment, which could be levodopa versus a new kind of levodopa, which typically is what's being compared, or it could be a placebo. Now, what's important about random assignment is that it's intended to make the groups equivalent on symptoms and other characteristics at the start of the study, before they get the intervention, but where they're being identified. So that if differences between groups are found at the end of the trial, we infer that the end of treatment differences between the groups were due to the groups getting different treatments, but not due to the groups being different at the beginning. Okay? Again, it is a way to make sure, or increase the probability, I should say, that what we see after getting treatment is a function of the treatments, the different treatments people were receiving, rather than the fact that they were different to start out with. And one of the problems with phase 2 studies, which are those smaller randomized controlled trials, is sometimes they're so small that the samples are really unusual and don't represent, typical people with Parkinson's, or don't relate to you personally, or the samples aren't even equivalent on symptoms at baseline. Sometimes that happens with a very small phase 2 study, that even that flip of the coin, like if you can imagine, when I teach statistics, which I'm not doing anymore, okay, when I used to teach biostatistics, you use the coin flip because you'd say, well, if you flip a coin 5 times and you have 3 heads and 2 tails, do you think the coin is biased? Probably not. But if you flipped it 100 times and you had that same kind of split of difference, you'd start to get a little more suspicious. So the larger the sample size, the more it is likely that random assignment will equate groups. But if the sample size is really small, sometimes it doesn't work. Random assignment doesn't equate groups on pre-treatment characteristics. Okay, and if possible, the studies are blinded. That means the patients don't know what treatment they're getting. Sometimes that's not possible, particularly with, let's say, exercise studies. Not always possible. Certainly the people doing assessments, if they're doing something like, you might be familiar with the UPDRS, which the part 3 of the UPDRS, which is the Uniform Parkinson's Disease Rating Scale. The part 3 is movement symptoms. And if your movement disorders doc or your neurologist does a standardized exam, they're probably scribbling on a piece of paper how you score on all these symptoms. Okay, and it would be really good, and it's usually the case, that in a randomized controlled trial, that at least the assessor is blind. Ideally, all the staff should be blind. except maybe for the statistician who has to assign people to a particular group. Okay, so just another moment to talk about what you find in a randomized controlled trial and what you might hear talked about. Statistical significance. Okay, you might see something like p less than 0.05. What does that mean? When you see that something is statistically significant, and there's a convention in science, nothing sacred, of using 0.05 as the cutting point for saying it's statistically significant, that means that the pattern of results or the group difference that observed are unlikely to occur by chance if the truth is that there's no active treatment difference. So we start with the assumption that sort of the default of these groups are equivalent, and you've got to present me with information that forces me to rethink it. Okay? And if it does, and if this kind of difference that we found is unlikely to occur less than 5 times out of 100, we'll say that it's statistically significant. But clinical significance is something different. And clinical significance requires statistical significance. That's when we're looking for meaningful changes that's perceptible and beneficial to patients in daily life and or to the clinicians, but mostly to patients, right? And so let's say for the UPDRS, that might be typically around 4 points to 5 points different in change. Okay, we're going to travel back to putting this in place with stem cell therapy. Now, the biggest excitement seems to come from very recent stem cell trials. These are the first in-human studies of these particular kinds of stem cells with looking at safety, side effects, best dose, and formulation here. Okay, now they are, I think it's fair to say, more advanced stem cell treatments are more advanced for PD than for any other central nervous system disorder. And recently, there's some phase 1 studies. Those are really those safety studies that have provided some hint of efficacy in the short term. And importantly, they didn't detect short-term adverse events. And you see in Nature, which is a major scientific journal, things like, and this is just April 2025, big leap for Parkinson's treatment. Symptoms improve in stem cell trials. What? This is a phase 1 study. What are you talking about symptoms improve? Well, really, these were safety studies. These are phase 1 studies. They're small. 2 studies combined 19 people. They all got either higher or lower doses of stem cells. And they got a lot of stem cells, all right? They might get, 1 million cells and with the hope that, 100 or 200,000 survive. And basically what they found, just because they do ask about symptoms. Some people got slightly better, others didn't get worse. So what's the problem with using these 19 people from phase 1 to make a conclusion about whether these things are going to help? The same kinds of problems with phase 1 studies as we have with personal testimonial and personal experience. What's causing this change? We don't have a comparator. We don't have a control group. We don't know whether this is really due to something other than the stem cell treatment, particularly when we know that more intensive interventions are more likely to produce larger placebo effects. And stem cell treatment is a major surgical intervention. So there's currently no approved cell therapy that can stop or reverse PD, and no stem cell therapy has been FDA approved for any indication. And what I think got people's attention is that there's a study that's claiming that it's now initiated, although they haven't started recruitment yet. It's a phase 3 randomized, sham-controlled, multi-site stem cell trial. They call it a phase 3 study, okay, which has never been one for stem cells, but there was no phase 2 study. So they never did even a smaller study, which compared people who got a sham versus active stem cell treatment. So even though it sounds like it was a big leap forward, it's really a labeling leap forward because there's a real push in the FDA for regenerative medicine. This is sort of a loophole where they allowed a phase 3 study without phase 2 results. I know I am running out of time. So anyway, I will say that 2 years is insufficient to make any conclusions about whether the stem cell therapy is effective and safe, because many of the side effects of stem cells take longer to show up. And I'm gonna just not go into this in detail at all, but this was a recent article that I ran across, which is Exercise Your Graft, which talks about the process of getting stem cells, differentiating them so they become dopaminergic neurons, implanting them in a person with Parkinson's. But they suggested, well, why not put them through a physical training intervention or a cognitive training intervention, because that helps neuroplasticity, or the way that the brain can change itself, okay, and might help to optimize the reinnervation of these dopaminergic neurons. I think that's a great idea. Hasn't been done yet, but I think it's a good one. Now here, which I think really important is, well, maybe I think it's important, is my opinion about what's happening with stem cells. Okay, I suspect that given the tremendous focus, it might become an option in the next 20 to 30 years. These things take a long time before we're sure they're safe. But if it becomes an option, it's going to be really expensive. I doubt that insurers are going to cover it. One estimate I saw that for a single patient, it's going to be somewhere around $800,000. And most importantly, it's not going to cure PD. Now that might surprise you, but PD involves loss of more than just dopaminergic neurons. People forget this, okay? They don't emphasize this at all. See this one, norepinephrine, these are all neurotransmitters, okay? Like dopamine, okay? And norepinephrine is found in the area of the brain called the locus coeruleus. By the time we start losing dopaminergic neurons, we have probably lost tons of noradrenergic neurons. That probably happens even beforehand, right? And so even if we could provide some replacement dopaminergic cells, neurons, we're still going to be shorting ourselves on all these other kinds of necessary chemicals that we need for our brain. I'm almost there. Well, I can quickly go through the biomarkers, okay? What's happening with biomarkers for diagnosis, like a blood test or skin swab? Well, why is it important? We know that people are sometimes diagnosed wrongly, and it certainly hurts clinical trials if 20% of the people who are enrolled don't really have Parkinson's. They're not going to be able to respond to a treatment. And there are brain scanning techniques like DAT scan. Maybe some of you have had DAT scan, maybe you've heard of it, which is a functional measure and requires exposure to radiation. They've gotten better, but they really don't allow you to see what's happening in the brain cells. It really is a functional measure. But we do know that by the time clinical symptoms occur, most of our dopaminergic neurons are lost. So if we can find a way to intervene early, prior to motor symptoms, we might have more success at reversing or slowing progression. And that's another reason why you might often see clinical trials saying we want people who haven't started medication yet. We want people who are within 1 or 2 years of diagnosis, because the idea is when you haven't lost too many neurons, you've got a better chance of turning things around. 15 years, forget it, okay? So why is a diagnostic biomarker test important? Again, more definitive diagnosis, early intervention might be more effective before symptoms are bad or even are evident at all at a motor level, and it'll improve clinical trial quality by ensuring that, I'm rusting myself, by ensuring that participants really have Parkinson's. And some of the ones that are out there right now, I myself have had a SYN1 test, which looks for phosphorylated alpha-synuclein. And we'll talk about what that is if I have time, okay? Which is a kind of abnormal protein that's found specifically in Parkinson's disease. and it folds to form these things called Lewy bodies, right, which are abnormal, which are associated with Parkinson's disease, and not all kinds of Parkinson's disease, but most kinds, okay? And they didn't even need FDA approval because it's just a lab test. It's really, it's a skin biopsy. And yeah, so they found out that I had this phosphorylated alpha-synuclein in 2 out of 3 sites, so I have Parkinson's, okay? And it's more accurate than DAT scan, okay? Even though, DAT scans have gotten better, that skin biopsies are less expensive, no radiation, and are more accurate than DAT scan. They also do something similar, looking for the same alpha-synuclein protein in lumbar punctures to get cerebrospinal fluid. But why do that if you can do it in the skin? I don't get it. Okay, and there are other new biomarker approaches which are still at a beginning stage. So one more thing I do want to talk about is.
Oh, Karen, can I just
Yes.
interrupt and say,
Sure.
take your time. You don't have to rush. We have all the time. This is on your
Oh.
time, and we appreciate it. So we are
Okay.
recording, and take your time.
Right, because I can talk fast. I'm from New York. Okay. So what should scientists be targeting for disease modification? Well, a favorite target, as I mentioned, are these Lewy bodies. So the neuropathologic hallmarks of progressive loss of dopaminergic neurons in the substantia nigra, which is a midbrain area, are accompanied by inclusions termed Lewy bodies. This is a Lewy body, kind of what it looks like, whatever it means, okay? But the main component of these Lewy bodies is this protein called alpha-synuclein, and it is a common disease modification target. And it is believed that this abnormal form of alpha-synuclein protein misfolds and forms these clusters called Lewy bodies. So much as in Alzheimer's research, you might have heard about people focusing on getting rid of the tau protein and real controversy about whether that's the appropriate target. We've got the same thing happening for disease modification in Parkinson's, where many new studies and new drugs are focusing on reducing levels of alpha-synuclein. And when I'm asked about the wisdom of this, I say, I don't think it's so smart, okay? And I think about the parable that many of you may have, or some of you may have heard of babies in the river, okay? If you don't know it, here it is, okay? So here's the parable. 2 people are walking by a river when they notice babies floating down the river. One starts grabbing the babies out of the river while the other one runs upstream. The first asks, where are you going? We have to save these babies. And the other replies, I'm going to see who's throwing babies in the river. So where do you intervene? Do you intervene at the end point, or do you try to intervene earlier? Okay, so we can stop the whole process from getting out of hand. So focus on initiating causes to achieve disease modification, not on the end product, not on the consequences. And I think that is a major mistake. And I won't go through all the possible causes that could lead to this protein aggregation of alpha-synucleins, but there's lots of them. So what intervention or treatment is most likely to lead to disease modification? Surprise! Exercise, exercise, exercise. Yay, yay, yay. Yay, PD Connect, okay? And exercise is unique because unlike drugs, which usually have a single target, exercise does so many different things at once. It improves brain volume, it promotes neuroplasticity, the way that the brain forms new connections. It reduces inflammation, it improves blood flow to the brain so we can get more oxygen and other nutrients that we need. It increases so-called neurotropic factors, which have to do with how the brain can remodel itself. You might have heard of BDNF, brain-derived neurotropic factor. There's GDNF, which looked promising in one study. And it improves the blood-brain barrier. And that blood-brain barrier is something that you might have heard about, which is sort of one of the problems with levodopa, because not much of it crosses through the blood-brain barrier. But that barrier is not so great in people with Parkinson's, but that barrier protects the brain from toxins and inflammation. So exercise helps that barrier. And to give you an example of a recent study that appeared just in 2024, a group in Yale, this is a small sample, but it's sort of proof of concept. They took a group of people who weren't exercising, small group, I think there was maybe 12, 14 people. They looked at their levels in a DAT scan of what do you call, how do I describe it? It's basically both the neuromelanin as well as the level of dopamine-related activity in the area of the brain where we lose dopaminergic neurons. And they looked at them both before they exercised and after 6 months of exercise. Typically, what one would expect over 6 months is that the levels decrease. In this exercise example, the average was shown by this. People actually increased their levels of dopamine, or this equivalent of dopamine. So again, small, and look, there was one person who did just the opposite, so it's not universal. Each one of these lines represents one person, okay? So although it might be statistically significant, it's promising. The idea that we can actually show changes in the brain or the functional parts of the brain, or functional image of the brain, to say that exercise, this is intensive exercise over 6 months, actually improved functional activity of remaining dopaminergic neurons in the substantia nigra. I think that's pretty impressive and very promising. And this will be my conclusion for now, unless people have other topics they want to talk about. And my recommendation is to practice at being skeptical about research claims. When I say skeptical, I'm not talking about radical skepticism, which is never accepting anything, like being a climate skeptic, which means denying everything that you hear, even in the face of evidence. I'm talking about scientific, moderate skepticism, meaning question logically with doubt. Don't reject everything, but seek evidence. And that's how you should form your conclusion. And I love this quote from Carl Sagan, because he says, what counts isn't what sounds plausible, not what we would like to believe, not what 1 or 2 witnesses claim, but only what's supported by hard evidence, rigorously and skeptically examined. This is my favorite sentence. Extraordinary claims require extraordinary evidence. And now I move you on to questions, and you can, you folks can figure out how you want to organize that. And just here's my contact information.
Wow.
So, yeah, I know it was a lot, and I really went fast at the end, so I apologize for that. Hopefully you could understand me.
Absolutely, we did.
Yes.
Thank
you
you.
so much.
Not too much.
We'll
My
leave,
pleasure.
yeah, you can leave that on for a second so people can write down your email address or take a photo or screenshot or whatever.
Sure.
Such a powerful presentation today, Professor. I'm going to call you Professor because
Please call me Karen, please. Please.
Karen.
We are peers here. With what you folks do at PD Connect, it's wonderful.
Thank you.
It's really wonderful. On the ground with large numbers of people, and in terms of also, you know, the Zoom access, there's nothing more important, because this is something that can really make a difference in people's lives, as opposed to some of the other stuff I talked about. Okay.
Right. Yeah. And if you want to stop sharing, then we can see everybody.
Oh,
We'll
okay.
just come back up here onto the
Okay.
screen here. And then here we are. Anyways, yeah, we do have some questions, and I think it's so interesting that, you know, here come the questions. One of the first questions was, you know, what about hyperbaric oxygen therapy, or, you know, heard anything about Dr. Abro's thermal tunnel in the brain procedure? Longevity supplements advertised on the Coast to Coast radio show. So stuff like that is going to come through, and
Saying that
yeah,
they cure Parkinson's.
cure Parkinson's. I mean, I know that.
What should I start with? Okay. Okay. If it's not published in a peer-reviewed journal, if it's just somebody touting their own expensive nutraceutical, watch out. Okay. If it costs money, and it costs a lot of money, it's probably not effective. All right. I mean, that's all I can say. If it costs a lot, it means less. All right. So that's just a general guideline to go by. It doesn't mean that effective treatments might not cost something, but if it's nothing that's backed by scientific evidence, and is just personal testimonials. I've talked to you about the problems with personal testimonials. Watch out.
Yep, we can go back. You can go back. If you just joined us late, you can always go back to rewatch this recording where she talks about some of those personal testimonials and things like that. You know, we do a lot of these questions are going to kind of go along those same things. But here's one, and this is one that it kind of maybe you can combine it. One is asking how important is diet, but the other one also asks, in addition to exercise, what else has helped you with your PD symptoms and progressions?
I can't tell you what you think, what I told you about personal testimonial. This would be the same, okay? But I will tell you that when I was first diagnosed, I had a pile of supplements, okay? And I didn't know if anything was helping. And especially when you take multiple supplements at once, you certainly, if you're feeling better, or you're feeling worse, you don't even know what's helping. I would say, given that there's really not great evidence at this point to support most of these supplements even crossing the blood-brain barrier in sufficient amounts, you really wonder how a single supplement could cause major changes. I remember actually one of the things that I was really hot on was curcumin, okay, which is also in a lot of Eastern cultures, has been used to treat lots of inflammatory conditions as well as here. But originally it was not in small enough molecular form that it crossed the blood-brain barrier. And I tried to get Cure Parkinson's, which is a UK-based charity, to get interested in doing a study on curcumin because they do repurposed drugs. And they were not interested because I didn't have an animal model, even though there are animal models out there that might support it, but that's what they required. So I do think you need to be really cautious about investing in a lot of money. I know as much as and I don't mean at all to criticize Dr. Mishley, she will offer you supplements. Some of them I still take, but most of them I do not, right? And can I tell you why one versus the other? To some degree, it's cost, okay? To some degree, it's the state of the evidence, okay? But, you know, I do think that we go overboard on looking for the magic supplement. And so exercise caution and keep your pocketbook tighter. Okay.
Great. I do see that Randy Phillips had his hand up. Randy, did you want to put your question in the chat or did you want to just unmute?
Can you hear me okay?
Yes.
Just a quick question. I'm a fairly new, been recently diagnosed. And the context that I've had with people who are diagnosed, the basic thing is everyone is desperate for some kind of treatment or cure, some magic bullet. And I appreciate your throwing cold water on that possibility. I know it sounds kind of a negative way of putting it, but really is what it is. The stem cell research is years away, if ever it's going to happen. But I'm wondering what you think with our current federal health leadership is running all that research money out the window to put toward defense and electric cars.
You want?
Go ahead. Yes, you got my question.
Yeah, I mean, again, this is not supposed to be, I guess, this is supposed to be politically neutral. I don't know how to stay politically neutral about this. As somebody who's been supported for 40 years by the NIH has been decimated. Okay, we used to be a country that could be proud of our scientific innovations. No more. It does when you disassemble an enterprise like the NIH and pull money from, let's say there's even a study that had been started that I understand was stopped, which is the Dan Korkos at Northwestern was doing a multi-site phase 3 study of intensive versus moderate intensity treadmill exercise. Great study, all kinds of biomarkers, really great study. Hard to recruit for because you had to take people without exercise. Funding was pulled. Okay, so again, when people have made an initial investment and there are ongoing longitudinal studies that some has been restored after there was public outcry, but essentially about 70% of Parkinson's money in the US has come historically from the NIH. You think about all these foundations and all their money, it's come from the NIH. That's the big funder of Parkinson's research. Was, not anymore. Okay, so I think that there's been a real destruction of science, and there's certainly not an impulse to support science by the current administration. So that's all I can say. And it's very upsetting to me to know that I'm just happy that I'm out of that game right now, but I have deep sorrow for my colleagues and my junior colleagues who are just starting out in this. No more science in this country, not the way, and it's not going to recover. It took too long to develop, and the infrastructure support is just not going to be there as well. And that infrastructure support was very important. Okay, and the critical review, I was involved in reviewing tons of studies. So they were first proposing to get rid of the peer review, and then they're just taking away the money on existing longitudinal studies. So it's just insanity. That's all I can say to you. Okay, and I'll shut up before yeah.
Let's move on. There are a lot of questions about something positive, which is your exercise regimen. Can you just talk to us about what you do or how you've done it? And
okay. Yeah, I mean, I started off focusing on probably running, cardiovascular activity, because it's high intensity. Okay, but I realized strength, and I think throughout my entire adult life, even though I was never an athlete, I always knew that healthy aging involves staying active, especially in midlife. So I just continued it to the point where I was, 50, 55, trekking to Everest Base Camp, and my 20-year-old son was afraid that I'd make him look like a fool because I was so cardiovascularly fit, and that was really important to me. And so I started with that and continue with that, although now I've reduced that to 6 days a week because I realize we do have to give our body, as it ages, time for healing. I've learned that somehow, although I haven't hurt myself badly. I mean, it's been okay. But added, I used to do, heavy weight training. And I went from heavy weight training into much more of compound weight training, where I'm hitting multiple functional movements like deadlift, not deadlifts, farmer's carries, things like that, where I'm using my muscles in ways that I don't know what the PTs have to say about this, but I like the idea of something where I'm employing multiple muscles at once in functional ways that replicate the way that we're going to need to use our bodies. Okay. Yeah, I could get big biceps, but if I were a bodybuilder, that's one thing. But for functional movement, I want to focus on functional exercise. And a lot of that is body weight, right? So that's another thing I do. For flexibility, I do hatha yoga. I've done that since I was a teenager, so I do that regularly. Also meditation. I put that under the exercise umbrella. We know that there's tremendous efficacy for that, and I'm trying to think what else I do. I rotate things all the time, so I'm not doing the same thing except for running, which is sort of a couple of days in a row, but it just builds on itself. So some days I'll maybe do less running, some days I'll do more running, more weight training, more, whatever. Probably missing some things, but if you name it, I do it. And I built it just as, time allowed, and 3 to 4 hours goes by very quickly.
We love that.
It sure does. Thank you for
Yes.
saying that. Teaching exercise, we are definitely advocates for exercise, and we really appreciate you telling us your regimen. But
all kinds of exercise and functional exercise. Loves the PT. Loves to hear that. A couple more questions. I know we've run over, but we really appreciate your time because it's way past dinner time on your side of the.
It's okay, but I have no appetite, so it doesn't matter.
Anyways, the question here is: you've been diagnosed for 15 years. What has changed, in your opinion, of finding a cure or a disease-modifying treatment from 2010 to 2025?
It's a great question, and I wish I could say. I would say 15 years ago, and actually I would say I'm going back 25 years because my dad was diagnosed probably about 10 years before me, so I was on top of the research literature. And the most encouraging thing that was coming out was exercise, even then, 25 years ago, was starting to come out. And that has been consistent. But just about everything else that looked initially promising from animal studies or from small trials, once you went into phase 3 trials, it didn't replicate. And I think the whole model of a single, what's different about exercise is you're hitting your brain up in so many different ways at once, okay? As opposed to what pharma's pushing, which is a single pill for a single target. We don't have one thing wrong with our brain. We have a system gone awry that eventually leads to maybe downstream these alpha-synuclein inclusions or whatever. But that's downstream out of a lot of things having gone wrong. Because the body, usually the brain has, we don't really understand enough about neuroscience, okay, about regular brain function, healthy brain function, and the aging brain. And obviously the biggest risk factor for Parkinson's is aging. Forget about genes. It is not much of a genetic condition, even though people ask you what gene is responsible for your PD. Most of us don't have any high risk, very high risk, very potent genetic risk factors. Few families there might be. I mean, my dad had Parkinson's, I had Parkinson's. Neither of us had, and we're both Ashkenazi Jews. Everybody said, oh, you should have a LAR2 variation. I don't. Neither does my dad. Okay. So, you know, I would say that I don't feel that unless there's I think we need a lot more basic neuroscience before we can understand normal brain function, to understand why things go awry. We have some hints at it, but we need to understand that better, and we need to develop interventions that are multi-tiered, okay, that target multiple things at once. Okay, and you know, am I more or less hopeful? I'm again, I am a scientific skeptic and have been that way for a very long time. So I had no rose-colored glasses on 25 years ago or 15 years ago, and I'm wearing the same glasses now.
Thank you so much. You know, just to summarize a little bit about your talk, I just love the way you told us and taught us about the different types of research and just sort of the, personal experiences and the placebo effect, and how, those are all real things, but then there's also real evidence about how it can be applied to everyone. So that's
right.
one take-home point that I got. And the other one was, I really appreciate that you talked about the timeline of research and that chart that you showed us between stage 1 and 2 and 3, and how long that takes before you get to anywhere, and how few, let's say, stage 3 trials there are. And then you find out, oops, on that one about how they didn't do a phase 2 at all.
Right.
That's very,
That's a very
they relabeled it.
And relabeled it. And I think the point that really hits home for me at the end, when you say, exercise has been shown, we're wearing our 10th anniversary PD Connect shirts, because 10 years ago when I started PD Connect, I knew I wasn't going to cure anything. And here we are 10 years later, and we still haven't cured PD. But in that time, I feel like we have healed people. We have helped them with treatment and have healed their souls, or their brokenness, or their depression, or their loneliness, or their freezing of gait, or whatever it is, their bradykinesia. And I feel like, as much as we love that there are big foundations and lots of money has to go to these research studies, I really appreciate the people that have supported PD Connect and all the other small, on the ground nonprofits or for-profits that can actually make a difference in the treatment and healing you right now. So
right now, and provide community and support at the same time as an intervention that we know is better than anything else out there right now. So let's go with what we know works.
Yeah,
absolutely.
And I love too that you talked about the whole babies in the river. I'd never heard that before, but my goodness, that is very powerful. Yeah, what is it called? barking up the wrong tree with this whole alpha-synuclein thing. Is that another way to say that? You know, and so everything is about destroying alpha-synuclein and all this money and all this time and research going towards alpha-synuclein. And what if it's not alpha-synuclein? That's the problem.
And if I had to put my money on it, it's not alpha-synuclein. You know, it's what happened that we have all this alpha-synuclein in our brain. And then they'll do studies where they find people who have just as much alpha-synuclein in their brain and they're asymptomatic.
Right.
Okay. So it is not, it's not a great target for intervention. And
Right.
yet that's what pharma looks at because it's something concrete.
Yeah.
Rather than clinician diagnosis or patient self-report, God forbid.
Right.
You know?
And I love too that you talked about how in your exercise regime, you do this, you do that, you do meditation,
all.
and you do it all. And one of the questions in the chat was about, well, what other neurotransmitters are being served? And you talked about that serotonin or the acetylcholine and the norepinephrine, and those are also being affected by exercise. So it's not that, you know, that one dopamine pill that you're taking is fixing everything, but the exercise, it seems, does get to address all those other neurotransmitters.
That's right. That's a very
Love
good
that.
point.
Thank you for putting that so clearly, that the exercise actually addresses all of those.
Yes.
I mean, because we talk about exercise all day, every day, but you know, it's really helpful for someone to come in, someone like you to come in, Professor Karen. Thank you so much for spending
my
extra
pleasure.
time with us here today. This was wonderful. And you know, if you don't know who we are, that's Lisa. I'm Steph. We're PD Connect. We teach exercise and education for people with Parkinson's. We're a nonprofit in the San Francisco Bay Area, but virtually wherever you are. You're also
That's
at
'cause,
our
you know,
speaker series.
virtually.
Yeah, because we're virtually here. But also you're at our speaker series, and this is being recorded, and this is going to be on YouTube, where also Dr. Daniel Quirkus was on twice before. So
yeah, if you
look
want to
back.
go
Yeah.
looking through those,
That is all free to you. We don't charge.
Mhm.
Please watch it. You can watch us on
Learn
double
about-
time.
And you can send them money too, folks.
Oh, well, thank you.
I mean,
Yes.
you know,
Thank
you guys need it, right? And then you
well.
worry about all the money that goes, these, you know, trials are so expensive and you're so wrong-headed even in their approach from the beginning. You know,
Well,
it's
thank
too
you.
many of them, you know, and we know the good that you guys are doing.
You're very
And
kind.
hopefully the folks that are listening know the good that it's doing for them.
Hopefully,
so-
hopefully. I think
I
they
think
do.
they do.
I think they do.
I think they
Thank
do.
you so very much for
Yeah.
spending all this
Pleasure.
extra time with us. We really appreciate you. Thank you to all of you who showed up here and show up for us every day.
Yeah.
We show up for you and supporting us in all the ways that you do. Thank you.
Folks, we
everybody.
are really lucky. Dr. or Professor Karen is just such a highly regarded person in the entire PD community. She sits on the councils of so many different foundations, and it just, her whole career has been devoted to understanding research and the right way to interpret it and follow it. And so we're just really grateful that you spent your evening with us today. So thank you again.
My
and keep
pleasure.
doing
My
what you
pleasure.
do.
Invite me to San Francisco sometime.
Yeah,
Wow.
We love that.
absolutely.
We love
Yeah.
that.
Okay.
Thank you. Thank
All
you.
All
right.
right. Thank you again for inviting me. Okay.
Awesome.
Okay.
Thank
All right.
you.
If anybody
Take
had
care.
any questions, if you have my email address, you can ask me a question directly because I respond to individuals all the time.
Great. Thank you so much.
All right. Thanks so much.
Take care,
care,
everybody.
everyone.
Okay. All right.